Hello, today's chapter is chapter 2, and this is going to deal with the chemical level of organization. Some of you may be thinking, chemistry, what's going on here? You cannot study biology without having a basic understanding of some basic chemical principles. They go hand in hand. In other words, one doesn't exist without the other. In biology, because it does involve a lot of chemical processes going on at the cellular level, which then translates to the whole organismal level, we're going to have to understand just a little bit of the basics. So don't panic. We'll try to make this as painless as possible. And this is certainly not taking a whole chemistry course and putting into one chapter. We're just going to hit on the basics to review for some of you who may have had chemistry, and for those of you who have never had chemistry, at least explain some of the background fundamental principles so that when we get into the anatomy and we talk about different physiological functions, that it will make sense because you have a basic understanding of some chemistry. Well, as always, we're going to have some definitions of words. So to start off with matter, matter is anything that occupies space and has some mass. It's going to be composed of elements. So what is an element? Well, that's a substance that cannot be broken down to other smaller substances by ordinary chemical means. Now we can break them down into smaller components, but it's not going to be easy. It's not by just standard ordinary means. So what are some of the most common elements? What we have found is that all living organisms are going to be composed of four main elements: carbon, hydrogen, oxygen, and nitrogen. And you'll notice in parentheses I do have the chemical abbreviation that we use that you would see, say, on the periodic table. When you go through this a lot, it just becomes second nature after a while that, oh, okay, carbon is C, and most of them are the first letters of the word, but then there's a few that we throw you for, such as sodium and iron, where we use Latin words. Am I expecting you to know the chemical abbreviations? No, because this is not a chemistry class. I always say I have to leave something for you to take in chemistry. So as long as you say, for carbon, if you want to write out the whole word, that's fine. If you want to just use C, that's fine. So those are elements, and there's over 100 elements right now. Oftentimes you hear us talk about compounds. So what's the difference between an element and a compound? A compound is when you have a combination of elements and they're in a particular ratio. If we were to look at, say, water, water is a compound. It is composed of hydrogen and oxygen. Well, it's in a very particular ratio, H2O, meaning for one oxygen and you would have two hydrogens. That ratio, you get water. Now, if you had two oxygens and two hydrogens, you don't have water anymore. That's hydrogen peroxide. So for the compound, it's in a very specified ratio, combination of elements. So a compound is larger than an element. This table is just showing the major elements and then some of the other elements that are found, but just not to as high a concentration as the carbon, oxygen, hydrogen, and nitrogen. So as I said, elements are going to be combined in a very fixed ratio to form compounds. And one thing to keep in mind is that compounds, when they're formed by these elements, they may now have very different characteristics from the elements that they're made up of. Kind of a classic example is naturally sodium is a solid at room temperature. Chlorine is a gas. It can also be under pressure put into liquid. Chlorine gas is poisonous. if you inhale it, it forms an acid, hydrochloric acid, in your lungs, and it will burn. So chlorine gas is poison. It can exist also as a liquid. You combine sodium and chlorine together in a 1-to-1 ratio, and you get sodium chloride, which is table salt. Now, an atom is the smallest unit of matter that will still retain the properties of that element. So if you have oxygen, the element oxygen can be broken down, or it is composed of atoms. Now, remember, an element you can't break down by ordinary means into smaller components. Is it made up of, for that element, is it made up of smaller components? Yes. It's made up of atoms. You have to do quite a bit of work to be able to detect the atoms at that level. They will still, an atom of oxygen still has the properties of oxygen. The atom itself can also be broken down into smaller components or building blocks. It's going to be very, very tough to distinguish. It took us a long time to be able to discover them. For purposes of this level biology, what are those subatomic particles? There's 3 that we will look at. There are more than that, but we're only looking at 3. Protons, neutrons, and electrons. So what's the differences between these subatomic particles? The proton has a positive charge, so in your mind think P, positive. It is in the nucleus. Now, please keep in mind, some of you may have had some biology experience and you hear the word nucleus in biology, and most people automatically think of the nucleus in the cell. That's not what I'm talking about. I'm talking about much, much smaller proportions. Nucleus just refers to something in the center. And so for subatomic particles, when I'm talking about the nucleus as a location, I'm talking about in the middle of the atom. So the proton is in the nucleus, or in the middle of the atom. It does have some weight to it. The electron has a negative charge to it. It is not in the nucleus. It's going to be in constant motion circling around that nucleus. It's very, very small. Now, when it's circling around the nucleus, depending on which book you pick up, and part of it depends on the time period, because we keep changing our minds as to what we call it. Some people will call it shells. Some people will call it orbits. Some people just refer to it as an electron cloud. Personally, I don't really care what name you want to give it as to where it's circling, as long as you know it is outside of the nucleus. The neutron has a neutral charge. It is in the nucleus along with the proton, and it also has some weight to it. This diagram is just showing a couple different ways that you can draw the electron to show, or the atom to show the subatomic particles. So this is showing, here would be the nucleus with this electron cloud around it. So here you have your neutrons, you have your protons, and then the electrons are going to be just somewhere out here floating around. In this type of model, once again, here is your nucleus with your protons and neutrons, and it's actually showing these orbits or shells with the electrons in them. Each element has an atomic number, and that refers to the number of protons. Now, normally the number of protons, which are positively charged, equals the number of electrons, which are negatively charged. So in most cases, that element's going to be neutral as far as the charge, because the positive and electrons cancel out. The mass number is equal to the number of protons plus the number of neutrons. And this is showing your periodic table. It is arranged in a very organized fashion, which we'll come back to after a little bit. Do you have to memorize it? By all means, no. But just know it is very organized. And if you ever take a chemistry class, this periodic table should become your best friend. because it tells you everything you need to know about how all the different elements are going to react. So these are all of the various, well, most of the elements, not all of them are shown. This has been updated since this one. Some of the newer elements, because not all of these exist naturally, only I believe it's 96 of them are naturally occurring elements. And so a lot of these newer ones here, the higher the number right here, the atomic number, tends to be the more recent ones, and a lot of those are man-made. Isotopes, this is a term where you have elements that have a different mass number. So they have the same number of protons and electrons, but they're going to have a different number of neutrons. They will behave identically in a reaction. So, as an example, carbon has several isotopes. Carbon normally has 6 protons, 6 neutrons, and 6 electrons. We refer to this as carbon-12. That would be the normal state. One of the isotopes is carbon-13. And in this case, it still has 6 protons, 6 electrons, but remember, in isotope, you have a different number of neutrons. So instead of having 6, it has 7. And then carbon-14, which is also an isotope, instead of having 6 neutrons, it has 8. Just so you know, carbon-14 specifically is a radioisotope, or a radioactive. You can use the full term. What does this mean? It means that the nucleus is going to start to decay spontaneously. As it decays, it gives off particles and energy. What type of energy is given off? It depends on that particular element. Carbon-14 gives off a low energy. At times, it can actually be even hard to pick up on a Geiger counter, which is the equipment you use to measure. Some isotopes give off a little bit stronger energy, and it just depends on which one you're working with. I have in the past, in research purposes, have worked with radioactive isotopes. You do have to have special training and certainly know all of the precautions to take when working with them. I have worked with carbon-14 a lot. The energy level that's given off is relatively low. You would be wearing your standard lab coat, gloves, etc. that I was wearing in my lab anyway, oftentimes just because I was working in a sterile lab. You don't want contamination, etc. Did I have to take extra precautions because I was working with C14 or carbon-14? No, I was obviously very careful when I worked with it. One of the things was certainly, well, I didn't want to spill anything, but what we call the half-life, how long it takes for it to decay, is the half-life. So let's just say you start with a strength of 100. The half-life would be how long does it take to get down to half of that, which would be, say, 50 units. And that varies by the element. For carbon-14, the half-life is just under 6,000 years. So I did not want to spill any of it, so I had to be very careful from that standpoint. Other radioisotopes, there are some where the half-life may be only 21 days. And so it depends on what you're working with. Some radioisotopes, such as P32, which is often used in medical diagnostic testing, in terms of working with it in a research lab, you're always working with a sheet of plexiglass in front of you because that is a stronger energy emitter. But it cannot go through wood, it cannot penetrate through plastic, so you would have a shield between you and where the sample was. I actually, many years ago, when I was at Oregon State University, was helping someone set up their radioactive isotope research lab, and they were working with one which is a very, very high energy emitter. It was one of those situations where it's like, yes, I have the background. I can help you set up this lab, show you how you maintain all your documentation. Every time you work with it, it must be documented. with any radioisotope, certainly with the one they were working with. Just to give you an idea how high energy theirs was, that was the only thing that was going to be worked with in this particular lab. They had a refrigerator. They stored their radioactive isotope sample in a metal container that was then sealed, placed inside of a refrigerator, which obviously then was closed. And when you walked in the room of that lab, if you had a Geiger counter, you could still pick up some low-level readings of that isotope, as it was not completely contained in there. So obviously, when they went to work with it, they had to use a lot of special techniques, equipment. And quite frankly, it was one of the things where I said, I'll help you set this up and I will show you how to do all the documentation. But when you're actually working with it, I don't think I want to be in the lab that day. There are benefits if you're on, well, if it's emitting energy, can that be harmful? Yes, it can. That's why you need to have special training. That's why you need to be aware of what the potential harmful effects are. So then why would you be working with it? Because there are some benefits to it as well. It will react the same way. I was working with it when I was doing research. And so it was a huge benefit for me to be able to track certain chemical reactions to determine the steps of a chemical process and what was going on. In terms of healthcare, living cells don't distinguish between the isotopes, so you can use radioactive isotopes as a tracer. And then it's very easy to detect it so that you can follow, like I said, through chemical reactions. The danger of it as this energy is being released, and it can break chemical bonds. It can cause abnormal bonds to form. You want to be especially careful with DNA that it does, because it can cause harm, and you don't want that obviously to happen. So it's one of those things, certainly from my personal experience in doing research, it was always a situation where you have to sit down and weigh what are the benefits versus, the adverse effects. What are the pros and cons for working with this? Are you going to get enough information that the benefits outweigh the potential harmful side effects? If not, then you might, don't do it. Think of a different way. The electrons, these are, remember, the negatively charged subatomic particles. They're floating around in those shells. The arrangement of the electrons is what determines the chemical properties of that element. And why is that? Because only the electrons are involved with the chemical reactions, not the protons, not the neutrons. They're in the middle of that atom. So think of it as they're being protected by the electrons, they're not being exposed. So it's only the outermost, and that's where the electrons are. As you increase the distance from the nucleus, energy will increase. And as I said, they're in constant motion. They are moving around that nucleus. So the number of electrons that are in the outermost shell will determine what those chemical properties are of that atom. If the outermost shell is filled, because each shell can have a maximum number of electrons, so that's designated. How many electrons can be held in each shell? If that outermost shell is filled to capacity, then that particular atom is going to be unreactive. It's filled. It's happy. Leave me alone. I'm full. We call those, typically they're gases. We call them the noble gases, the inert gases, or the stable gases. It all means the same thing. It just depends on which book you use. And like it's another case where over time somebody comes along and says, oh, this has been called the noble gases. Well, let's change it now to the stable gases or inert. Why they change it, I don't know, but they do. It's probably to confuse students. If the outermost shell is not filled completely, then it will be reactive. So how many electrons can be in the outermost shell? Well, the innermost shell right next to the nucleus, that one can only hold 2 electrons. The second shell can hold 8 electrons, and the third shell can also hold 8 electrons. So once again, I bring this periodic table up to show you that it's going to tell you everything that you need to know. You will notice that the periodic table, you've got your rows, you've got your columns. The way it is designed, if you look at the rows, which we call periods, but just think of it as rows. In chemistry, they'll call them periods, but they're rows. Essentially, what that is telling you is how many shells you have. So up here in one, this row across, they only have 1 shell. In row 2, they've got that innermost shell, which I just said a bit ago could hold 2 electrons. Once that's filled, now you have to move to the next shell, and you're moving from inner to out. So this would have this first shell, but now the outermost shell is going to be the second level. Third row, it's going to have 3 shells. Fourth row has 4 shells. So what do the columns mean? The columns tell you how many electrons are in that outermost shell. So if you look, they call them groups, but if you look in column 1, that means there's 1 electron in the outermost shell. It's not telling you the total. The total is going to be a number here. We're not going to worry about that right now. It's just going to show you in the outermost shell. Column 1 has 1 electron. Column 2, there are 2 electrons in the outermost shell. These react differently and play by different rules here. And we're in biology, we're not going to be dealing with these elements as much, and we're certainly not going to be dealing with these as much. So for biology, we're going to be dealing mostly up in this area and then also here in this area in terms of the elements. So over here, if you look here, this is column 5. Well, guess what? There's 5 electrons in the outermost shell. Now over here in column 8, most of these have 8 except for helium, which is right here because it's only in the first row, and the maximum is 2 electrons in its outermost shell. But in column 8 here, all of these are filled to their maximum capacity. So these are the inert gases or the stable gases or the noble gases, whichever name you want to give. These are unreactive. That's an example. Like this is Ne is neon. That's why they use neon a lot as a gas, like signs, etc. It's very unreactive, so they don't have to worry about it being, say, explosive. Helium, same thing. Used, like, say, for balloons, you use helium gas. We don't use hydrogen gas anymore. Helium is unreactive. Hydrogen, totally another story. So once again, that periodic table will tell you most of what you need to know. So just, if you're ever asked, well, how many electrons are in the outermost shell, how reactive is it going to be, just look. Where is its placement in the periodic table? So like right here is carbon. Carbon's in column 4. So it means that there are 4 electrons in its outermost shell. If you're looking at carbon, you're only dealing with the electrons in the outermost shell because those are the ones that are involved with chemical reactions. You're not worried about the inner orbits from that. So to form chemical bonds between various atoms, what you're looking at is what is the interaction of those electrons just in that outermost shell. Those interactions are going to cause or form the bonds. What do I mean by that? It means they're either going to be sharing electrons, and when they share them, it's usually a pair of electrons that they share. They may donate electrons from one to another, or they may be received. If somebody donates it, somebody else is going to receive it. Just a quick disclaimer here, I am working from home and I do have a cat. And so sometimes you may hear some background noises of my cat as she decides she doesn't want to be in the office anymore. So I do apologize for that. So the different types of bonds, there's three main ones that we're going to look at, once again, as it relates to biology. So I'm kind of filtering the chemistry in terms of, if some of you have had chemistry or maybe physics even, you might be thinking, well, I've heard of some other type of stuff. I'm only going to be presenting as it relates to biology. So there's three different types of bonds that we're going to look at: ionic, covalent, and hydrogen. We're going to talk about each one of them. Ionic bonds are, in terms of strength, at the intermediate level. Ions are atoms or molecules that have an electrical charge to them. Why do they have an electrical charge? Because they either lost an electron or they gained an electron. Remember, electrons have a negative charge. So when you lose an electron, if you normally have the same number of electrons and protons, and you lose an electron, so you lose one negative charge, you had equal positive, equal negative, you lost a negative, so now you have one extra positive charge, if you will, that's going to give you a positive charge. If you gained an electron, that's going to give you a negative charge. Well, opposite charges attract. And so ionic bonds are formed due to the attraction between these opposite charges. How did these opposite charges form? Because of the gain or loss of electrons. So in this diagram, it is showing sodium was in column 1. So that means it has one electron in its outermost shell. Remember, we don't care about the innermost shells because it's only the electrons in the outermost shell that are involved with chemical reactions. So it has one little lone electron here in its outermost shell. It needs to fill to 8. Well, that's pretty kind of tough to go and find 7 more electrons to stick there. So what is it going to have to do? Well, okay, it's easier just to get rid of it. So let's get rid of it. So it loses that one electron. It becomes positively charged. Chlorine, on the other hand, over here, it has 7 electrons in its outermost shell. How do you know that? Well, if you look on the periodic table, it's in column 7. Is it going to lose 7 electrons? No, it needs 8 to fill that outermost shell. So just pick up an electron from somewhere. So sodium gets rid of an electron, chlorine gains an electron. So now chlorine, because it gained an electron, becomes negatively charged. Opposite charges attract. So positive, negative, they attract. They form sodium chloride. It's an attraction between those positive and negative charges. That is an ionic bond. Covalent bonds are formed where instead of somebody giving up and somebody else gaining an electron, you're actually sharing them. These tend to be the strongest of the bonds that we talk about. And you can share, usually, like I say, it'd be pairs of electrons. You can share one pair. That would be a single covalent bond. Sometimes you'll see where they'll actually share 2 pairs of electrons, and that's a double covalent bond. Not quite as often, but it is possible. There are instances where you will share 3 pairs, and that would be a triple covalent bond. Polarity refers to the fact that, yes, you can be sharing, but as we all know, sometimes that sharing is not quite equal. Kind of like if somebody comes up and asks you to share something. Somebody comes up and asks me to share my Hershey's candy bar with almonds in it. I might have to think a little bit about that as to whether I'm going to share equally. So, polarity is referring to the sharing of electrons. If it's nonpolar, and you'll often see something written as a nonpolar covalent bond, that means that there's equal sharing. You hit me on a good day, I'm going to split that candy bar in half. Polar, and you would often see this either as a polar bond or usually as a polar covalent bond, because it is, regardless, either one of these will be covalent bonds because you're sharing. Polar covalent bonds are when it's an unequal sharing. You hit me on a day when I really needed my chocolate, and I might share but and break off a piece for you, but I'm not going to guarantee that I'm breaking that evenly. So with the polar covalent, you're not sharing those electrons equally. If you're not sharing the electrons equally and they have a slight negative charge electrons to them, that means you're going to end up with regional differences in charges. So in other words, what's going to happen here, this is shown with water. So this red up here is the oxygen atom, and the slightly gray are the hydrogen. Oxygen is bigger than hydrogen. So you're going to share a pair of electrons here, and you are sharing a pair of electrons here. Oxygen is bigger, so when it's sharing, okay, it's going to share with the little old hydrogen here, but it's going to be pulling those electrons closer to the nucleus of the oxygen. Think of tug of war. You've got my son pulling this way. You've got my younger daughter who's a little tiny thing over here. It's not going to be equal. She's tug of war. She's not going to win. Sorry. He'll be nice and not take it completely away, but it's going to be pulled closer to the larger atom. So these electrons are negatively charged and they're being pulled towards the oxygen. That means over here, the electrons aren't here because they're being shared on the other side. You end with a slightly positive charge over here, and on the opposite end of the oxygen, it's slightly negative. So you can see this drawn down here where the hydrogen's over here, there's a slight positive and there's a slight negative over here on the oxygen. Why? Because it's yes, it's a covalent bond, but it's polar. It's not being shared equally. So a molecule, oftentimes this terminology that we use, is just where you have two or more atoms that are combined together. That could be two oxygen atoms. It could be two hydrogen atoms. Oxygen in the atmosphere is often what we call O2. It's two oxygen atoms that are combined together. A compound, and oftentimes people say, okay, well, then what's the difference between a molecule and a compound? A compound is when you have two or more atoms of different elements. So technically O2, you could say that is a molecule. Now, when you have H2O, then that is a compound because you have both oxygen and hydrogen in there. So our third type of bond is a hydrogen bond. So we had the covalent bonds that are the strongest, ionic bonds are intermediate in terms of strength, and then the hydrogen bonds are going to be the weakest of the three types of bonds. Just because they're weak doesn't mean, oh, you can ignore them. No, they are very, very important in helping to determine overall structure, like overall three-dimensional structure of proteins. They're very important with DNA structure, etc. So they're very important, even though they're weak. Where are they found? They're often found sometimes within one large molecule, sometimes between molecules. And how are they formed? Well, when you have these polar covalent bonds, and that has to be formed first before you can have a hydrogen bond. Remember how we just said you've got those partial positive and partial negative regions? Opposite charges attract. And so what happens is, as we just saw with water, you've got your oxygen here, your hydrogen here, because of the polar covalent and unequal sharing. This area right here along the red is going to be slightly negative on all of these separate molecules here. So when you form the compound of water, you've got a negative charge here, but all the hydrogens have a slightly positive charge because of the polar covalent bond. Well, this is slightly positive and this is slightly negative. Opposite charges attract. So a hydrogen bond is formed between those slightly positive, slightly negative areas. Why were they formed? Because you had a polar covalent bond. Hydrogen bonds are very weak. Just by tradition, they tend to be drawn as kind of a slashed or a dotted line to indicate that they are there, but they are weaker. So when you are looking at your water bottle, yes, water is H2O, meaning you've got two hydrogens to one oxygen. That is the ratio that this compound, because you've got two separate elements, they are combined in this particular ratio. So this would be forming your water compound here. Now, in your water bottle, do you just have one of these? No, there's millions and millions of these molecules, and they're held together by the hydrogen bonds. For chemical reactions, first, we're going to be looking at energy. Do you just understand this? There's two types of energy, kinetic energy and potential energy. Once again, all of this is relative to biology. Kinetic energy is the energy of motion. Potential energy is that stored energy. And so what we're looking at in terms of biology, when you're talking about stored energy, where is it stored? It is stored in those chemical bonds. It's stored in those ionic bonds or covalent bonds or hydrogen bonds. And whenever you break a bond, you will release that chemical energy, that potential energy that is stored there. You're going to release that energy, and then the cell will use that energy to carry out other types of reactions. So you can have, in terms of the forms of energy, you can have mechanical energy, which involves the movement of matter. You have radiant energy, which is like ultraviolet light, where it's transmitted as waves. The energy is emitted, it's transmitted as waves. And then you can have electrical energy, which is going to be supplied by electrolytes, so charged particles. When we talk about chemical reactions, reactants, which are also sometimes referred to as substrates, that's the starting material. And then what you end up with is your product. Something to keep in mind is that some basic laws of chemistry, you can neither create nor destroy matter. You can rearrange it all you want, but you don't magically create and you don't destroy it. It doesn't disappear. You can rearrange it, but once again, you can neither create nor destroy it. When you're rearranging it, what are you doing? You're making bonds, you're breaking bonds, you're shuffling things around. You must always be able to balance chemical equations. Now I'm saying this just so that you know that when we look at chemical reactions and we break bonds and we make new ones and we're rearranging all the carbons and the hydrogens and oxygens and sodiums and potassiums, etc. As the first law states, you can't have it magically disappear. So if you start with, say, glucose has 6 carbons, at the very end of all this rearranging that you're doing, you still better have 6 carbons. And that's what we're talking about balancing, is that whatever you start with, you've got to end up with the same number. You start with 20 hydrogens, you better have 20 hydrogens at the end. It may be in a totally different combination as you rearrange it, but you have to have the same. Now, am I going to be asking you specifically, say, here's some substrates, here's the final products, and ask you to write out chemical equations? No, I will not. Will I be asking you to balance chemical equations? No, I will not. I just want you to understand that we can't have things disappearing on us. There's different types of chemical reactions. Synthesis reactions just means you're building up compounds. So here you're starting with 2 separate components. I'm just drawing it as A and B, and you're going to combine them. You're going to form a bond between them. So now you're going to store energy in this bond. So you're going from 2 small things to 1 large thing. Decomposition reactions is the reverse of that. You're breaking down compounds. So you're starting with a larger compound and you're going to break it into smaller components of it. Anytime you break a bond, you're going to be releasing energy. Anytime you form a bond, it requires energy. Oftentimes, and this is more common in biology, what we will see is that you're doing a combination. You're breaking and you're building compounds. So you're shuffling things around once again. Remember I said you're rearranging things. So you're going to break a bond between A and B, and now you're going to, sorry, B, you're off by yourself. Now we're going to combine A with C. Very broadly, compounds can be organized as either inorganic versus organic. Inorganic compounds do not contain both carbon and hydrogen. Examples would be carbon dioxide, water. Water, as we've already seen, H2O. Yes, it's got hydrogen, but it doesn't have carbon in it. There's a couple very few exceptions to this. Organic compounds, which is what we're going to spend most of our time looking at, contains both carbon and hydrogen. This would include things like your sugars, your proteins, your lipids. Water makes life possible. Most organisms are at least 65% water, so that always brings the question up, if water is present, is life present? This is one of the reasons why, as we expand and explore different areas, if you're curious whether life may have existed there as we know it, and obviously first time you look at things, you first start looking at things that we are familiar with. So as we go to explore, as an example, on Mars, and the question is, did life ever exist on Mars? Well, we're going to look for life as we are familiar with, as we know it. And one of the basic things is that most organisms are dependent upon water. So that's why they're looking to see if water was present on Mars. If water was there, then that might give us a clue of, could it have supported life as we know it? And so that's indeed one of the things that they are looking at. Just as a side note, it's very interesting for me in that I have a son that just recently received his master's degree in geology, and he's always been fascinated with geology and also astronomy, and he initially thought, oh, I want to study dinosaurs. And as he started college, he was introduced to someone, and he actually now, his master's degree, which he just got, and he's continuing with his PhD, he's very involved with research supported by NASA. And ultimately, he's with a group of people that are studying the chemical makeup of the rocks and the clay on Mars to determine what is the chemical makeup of the geology on Mars, and then that translates to, depending on what they find, could it potentially have supported life on Mars? So I tease in that in a roundabout way. He is studying biology. But it directly deals with this, is if water was present, could it have supported life? Water has some very unique properties to it that allows it to support life. And like I say, it helps with the whole issue that life is definitely dependent on water. And what are some of these properties? Well, it acts as a lubricant. It helps to cushion things. It helps with temperature control. It helps as a solute, meaning a lot of things can be dissolved in water. The hydrogen bonds that are found with water is responsible for water's ability to have some of these unique characteristics. And one big thing is to resist temperature change. Because water is able to resist temperature change, that what I'm talking about is you can have, and all of you are actually familiar with this, not that any of us think about it, but you're tired, maybe you're cold in the morning and you just want a cup of coffee, and you fill up, let's say if you have a a coffee pot and you fill it with water and you heat it. And you're like, but I want my coffee now. I want that water hot now. But you have to wait for it to heat up. That's an example of you're applying heat to that water. Well, the water, it takes a long time, and it always seems to be longer. The more you want it, the longer it takes for it to heat up. That's because it's resisting that temperature change. And you might find that very aggravating. However, having the water being able to kind of moderate the temperature, it allows life. Because think about it. I'm recording this in the summer where it's been hot. Personally, the hottest temperature I've ever been at was 119 degrees. Does that mean that my internal body temperature was 119? No, because I wouldn't be here if it was. If my body is over 65% water, that water is able, in the body, is able to absorb a lot of that heat and resist the internal temperature from increasing. So it's just a unique property that water has to be able to absorb a lot of heat without seeing your internal body temperature increase. When water evaporates, it's going to take some of that heat with it. And that's what we call evaporative cooling. How does this translate to you? Well, whenever you sweat, as annoying and uncomfortable and unpleasant as that may seem to you, that is your body's way of trying to cool you off. Because as you sweat, it's taking that heat away. And you may not be aware of it in a humid climate quite as much. Just all you know is you're sweating a lot. When I lived in New Mexico, I was there for over 20 years. It's very, very dry there. Humidity there, people don't know what humidity is there. They complain when it's 20% humidity. Oh, it's so humid. When you're out doing things, there's a drawback of it being dry like that. It seems cooler because as soon as you're sweating, it's pulling that heat away and you do feel this cooler. 90 degrees and 20% humidity feels a lot cooler than 90 degrees and 95% humidity. The drawback from a healthcare standpoint is when it's that dry and you're sweating, it immediately is evaporating, so you're not aware as to how much you're sweating, which translates to you are not aware as to how much water loss you are having, and it's very easy to become dehydrated. From the body standpoint, it's cooling. So that's a unique thing that water has. The other thing unique, and there are many unique characteristics of water, is that a lot of things can be dissolved in it. A solution is a liquid that has a very uniform mixture of two or more substances in it. It's going to contain both the solute and the solvent. And so you're going to be dissolving something in it. So something does the dissolving. And when it's equally mixed, you cannot distinguish between the two. So the solvent is a dissolving agent, like water. The solute is something that's being dissolved, salt. Or maybe you're familiar with if you make Kool-Aid. The Kool-Aid powder is the solute, water is the solvent. An aqueous solution is simply when water is the solvent, because anything could be the dissolving agent. Now, when we are talking about human biology and generally biology, in general for any animals, water is usually the solvent. So that's called an aqueous solution. So why is water such a great solvent? Because of its polarity, because of the unequal sharing of covalent bonds. You start to see everything comes back around. You're probably thinking, oh my gosh, we're jumping all over the place. It all comes back kind of in a circle. Water is going to come around and separate and surround ions when you have a solution. In other words, you take that sodium chloride that we've talked about before, the table salt, where you formed, remember, ionic bonds here. Typically what happens when you take a crystal of something that's held together by ionic bonds and you put it into water, into the solvent, what's going to happen? It's what we call dissociation. The charges that are holding here, the water is going to come between them and separate them out. This is a good thing. Why? Because, just to give you an example of why it's important to know that with these salts, that when you put them in water, they're going to dissolve and water is going to surround them. And the reason that's important is because you need a lot of these components for proper functioning of the body. You need sodium, you need chlorine. So when you intake sodium chloride, when you eat something that's salty, you get the salts. Can this large crystal pass into an individual cell? No, it cannot. It's way too big. This is huge when we're talking at the cellular level. There's no way this is coming through. So what do you have to do? Excuse me. Because, once again, here we go. We're over 60% water. This is going to dissolve when it's inside you into the watery solution. This small little sodium is so much smaller than this huge crystal. The chlorine is so much smaller than this huge crystal. This can now be passed into the cell. So you intake it as this, dissolve it in the water when it's in you, and now you can utilize this as needed. Hydrogen ions, hydrogen with a plus charge, hydroxide ion, which is oxygen and hydrogen in 1-to-1 ratio, and it has a negative charge to it. Both of these are very reactive. There are a lot of different definitions from a chemistry standpoint of what an acid and a base is. As far as we are concerned, these are the definitions we're going to use for this class. An acid is a compound that's going to release or donate a hydrogen ion in a solution. So the hydrogen ion concentration, the brackets refer to concentration if you're not familiar with that. So the hydrogen ion concentration is greater than the hydroxide ion concentration. The pH is going to be less than 7. That is what an acid is for us. For a base, we're going to talk about a compound that accepts the hydrogen. So the hydrogen ion, because it's being accepted and bonding, that concentration is less than the hydroxide ion concentration. The pH is greater than 7. Your pH scale is a scale that goes from 0 to 14. 7 is neutral. Buffers are substances that you have a solution and it has a buffer in it. You can add either an acid or a base, and that buffer is going to help minimize any change in the pH. It prevents it from altering. It'll change a little bit, but not drastically. Here's an example of the pH scale and different common substances. So as you can see, 7 is neutral. As we just said a bit ago, anything less than 7 is an acid. Anything higher than 7 is a base or an alkaline. Base and alkaline are the same thing. Notice some of your common everyday substances. Coffee is pH 5. Some of your juices, look at this. Grapefruit juice, about 3. Some of your sodas are 3. pH 2, your stomach, the gastric juices or the juices in the stomach, it's a pH 2. That is very, very acidic. That's one of the reasons why, I don't mean to gross you out, but one of the reasons why when you are sick, when you throw up, it burns as it's coming up. Why? Because your stomach's at pH 2. Those juices, that fluid, is very acidic. A lot of people realize that when you are in this range, pH, even 3, 2, and certainly 1, you can get very severe burns. Anybody that works with strong acids, anyone, a mechanic like battery acids, knows these can cause very severe burns. What a lot of people forget is that at a very high pH, very strong basic solutions can also cause very severe burns. And that's one of the reasons why, look what some of your cleaning things, bleach, uh, ammonia, a lot of the cleaning solutions are very, very high pH. And that's one of the reasons why on the labels they will say to wear gloves. Those warning labels that nobody reads. There's a reason why it says to wear gloves. It's to protect your hands because you can end up with severe burns. So our life is dependent on water. So any type of contamination of your water source, that can pose some very serious environmental threats, very serious health threats. When I teach microbiology and we get into specific diseases and how they're transmitted, so many things can be simply prevented if you have a clean drinking water source. And all of us have seen that whenever there's been disasters, whether it's hurricanes, fires, or get a clean drinking water supply is going to be one of the first and foremost things that you need to do. Number one, people need water to survive. Number two, to help them prevent spread of disease and infections. We look at different types of biological molecules, and this will be looking at your organic molecules. So we're kind of shifting now gears a little bit to look at these. The four main classes. They're divided into these four groups: carbohydrates, lipids, proteins, and nucleic acids. So we're going to look at all of these. For all of them, just keep in mind that we use the terms monomers and polymers. The monomer is going to be the building block, kind of repeating units that make up the polymer. So the polymer is a large substance that tends to be made up of identical little building blocks. Think of like you're building a big old wall, brick wall. Each individual brick would be the monomer, but the entire wall is the polymer. So we're going to start with carbohydrates. Basically, these are sugars. There's different types of sugars. The way we classify them: monosaccharides, polysaccharides. Monosaccharides tend to have, they're smaller, as the name implies. They tend to have a certain ratio. For every one carbon, there would be two hydrogens and then one oxygen. Carbohydrates tend, the naming of it, there's a specific way that you name them, but they tend to end with OSE. So you see something that ends with ose, that's going to be a carbohydrate. Sucrose, maltose, lactose, all of those will be your carbohydrates. Monosaccharides, this is going to be the main source of energy for us at the cellular level. When they're in an aqueous solution, they tend to form ring structures. Two main types that we talk about in biology. We have pentoses, which are a sugar that contains 5 carbons, and then hexoses contains 6 carbons. And this just shows you an example of some of the common monosaccharides. As I said, they will be in a ring structure. When they're in an aqueous solution, you take them out of the water solution, and typically what's going to happen is you'll have one of these bonds break, and then it extends out into basically it looks like a long line. So on the top here, glucose is probably your most common sugar. Certainly it's the #1 choice for cells to use to break down for energy. And it is a 6 carbon sugar. Just FYI, if you're trying to count the sugars, each one of these points, if it's not marked there, that is a carbon. So you have 1, 2, 3, 4, 5, 6. And then both of these down here are your pentoses: deoxyribose and ribose. Do you have to know the structures? No, you do not. Disaccharides are when you take 2 monosaccharides and you combine them together. The most common examples are maltose, where you take 2 glucose molecules, combine them together. Sucrose is glucose and fructose, and then lactose is glucose and galactose. Some of you may be familiar with lactose, and that is commonly referred to as milk sugar. Just kind of FYI, if you are lactose intolerant, that means you do not have the enzyme that is necessary to take lactose and break it down into these two smaller monosaccharides. And the body has a hard time, it cannot utilize the lactose, so that's going to cause some, we'll just say, some digestive issues. There are ways around it. If you buy lactose-free milk, usually what they've done is they've added the enzyme and it already breaks it down for you. And so once again, that's just showing the chemical structure of these disaccharides. Polysaccharides. Monosaccharide is when you have just one of the components. Disaccharide is when you put two of them together. Polysaccharides, you just have a long chain of monosaccharides linked together. We see this oftentimes as storage molecules and as also structural compounds. Some of your different types of polysaccharides. Starch. This is found in plants as a storage mechanism. Starch is essentially in long chains of glucose. So as the plant makes more glucose, it just keeps adding, another glucose at the end of the chain. If it needs glucose, then it can just start breaking it off the chain. Animals do not make starch. Instead, we make glycogen, but it's a similar type of strategy. It's that when there's excess glucose, you add it on the end of a chain. When you need glucose, when you're short and you need, then you go to the glucose storage savings account and pull some out. You pull it out one at a time. The difference structurally is starch typically is long chains, and glycogen has a lot more branching to it. Cellulose is another type of polysaccharide found in plants. It's a structural component. Cellulose is the most abundant organic compound on Earth. And then chitin is another type of polysaccharide. That forms that hard structural compound like the exoskeleton of insects, crustaceans. It's also found in the cell wall of fungi. And this is just showing you how you have these, each one of these is an individual glucose. So you have these large structural compounds, and you just add a glucose. When you have extra, just add it all in the end and just have these continuing chains growing longer and longer. And then when you need glucose, you just start breaking off one at a time. At the cellular level, when the cell needs energy, its first choice is always going to go for a carbohydrate first. That's the easiest thing for it to use. Lipids are your fats. So this is your second group of molecules, of organic molecules. The fats, this is going to be composed of compounds where you have carbon and hydrogen. They're linked by nonpolar covalent bonds, so there's equal sharing. Lipids have a characteristic that they are always going to be hydrophobic. That means they do not want to mix with water. They don't want anything to do with water. Water and oil do not mix. That is true. So they don't want anything to do with it. Just so you know, in terms of energy usage, like I just said, the cell's going to use carbohydrates first. At the cellular level, if carbohydrates are not available, then it will go to lipids for energy source. So they are often used as energy storage. Another huge function of lipids is that they will help to cushion a lot of the vital organs, and then it also helps to insulate the body. Oils are your liquid fats. Triglycerides are a type of lipid. Contains glycerol, which is a 3-carbon compound, and then 3 fatty acids. So this is just showing up here the glycerol component. So there's 3 carbons here, and then each one of these chains is what we call fatty acid. And this is showing specifically an unsaturated because you have a double bond here. You could have had another hydrogen here, On this side, another hydrogen here. So you do not have the maximum number of hydrogens there. Essentially, you have a double bond here. What that does, instead of this third chain lying straight and parallel, it puts a little kink in it. And so that's called an unsaturated fatty acid. So this is just showing here once again to form the triglycerides. You have the glycerol, you have the 3 fatty acid chains that become attached to it. Now, oftentimes people from a nutritional standpoint may be saying, ooh, triglycerides, those are bad. I have to be tested for those a lot. It's one of those things. You need to have a certain amount, but a lot of us have way too much. Once again, unsaturated means that there's a double bond in there. You have less than the maximum number of hydrogens possible. Saturated is where you do have the maximum number. There's no double bonds. They're going to lie all nice and straight against each other. Phospholipids. This is a major component in the structure of cell membranes. It helps to form what we call a bilayer. You have 2 layers of phospholipids, and there's a hydrophobic area and there's a hydrophilic area. So what in the heck is a phospholipid? It contains a glycerol, just like we just saw, that little 3-carbon chain. Now, triglycerides have 3 fatty acid chains. Phospholipids only have 2. And instead of the 3rd fatty acid chain, it's replaced with a phosphate group. So this is what it looks like. Here is your glycerol right here, your 3 carbons. Here's a phosphate group. Remember, a triglyceride, that's where your 3rd chain would be. But instead, with phospholipid, we put a phosphate group here. And then we have, here's our 2 fatty acid chains. They may be saturated, they may be unsaturated. Just so you know, with phospholipids, we tend to oftentimes refer to them with the very highly technical terms of this area as the head and this area as the tail. One reason for distinguishing that is that this head region, the glycerol and the phosphate group on it, is hydrophilic, meaning it does like water. It will dissolve in water. The tails, once again, are hydrophobic. And that becomes very important. Why? Because in our membranes, the cell membranes, and even within the cell, you're going to see some of the membranes of the organelles within there. But certainly your cell membrane, so having inside the cell versus outside the cell, these phospholipids form the membrane. That's what it's comprised of. And it's a bilayer. Well, the tails that are hydrophobic tend to come always just naturally and fold in towards each other. So what this does is outside the cell is hydrophilic. It likes water. Inside the cell is hydrophilic. It likes water because remember the heads like water. But this region in the middle where the tails are is hydrophobic. It does not like water. What does that mean? As we see when we study the cell, that's going to help regulate what can pass across the membrane. Why? Because of the chemical properties of it. Why? Because those tails are hydrophobic. They don't like anything that's dissolved in water. They don't like water. They don't want to be around it. Steroids are another group of lipids. They are a larger compound. They're actually a ring structure. There's 4 rings that are fused together as your basic template. Cholesterol is an example. It's very common in animal cell membranes. Cholesterol is used as a template for making other steroids. So once again, kind of like triglycerides, cholesterol often gets a bad rap that, oh, it's bad. Well, you need to have some. Why? Because it's a common component, structural component in the cell membrane. It helps to stabilize it. Cholesterol also is used as a template for making other steroids. And if you're thinking, well, what other steroids are there? Hormones. Hormones are either protein-based or steroid-based. And so to make some of the other steroids, you have to have the cholesterol. So a little bit, you need to have some, but once again, too much is a bad thing. So this is what the basic structure of cholesterol looks like. So like I was saying, for the steroids, you've got these 4 rings, and then the differences are what you add to it. Protein. So you've got your carbohydrates, you've got your lipids. Now we're moving on to the proteins. Proteins, there are a lot of different functions of proteins, a lot of different types of proteins found. They are essential to most functions required for life. The building blocks are amino acids. Proteins are going to have a unique shape, this 3D shape, and that's going to give it a very specific function. Structure determines functions. So proteins are going to be involved in almost every single activity in the cell because they're involved with chemical activities, they're involved with structural activities, etc. So we look at amino acids. They're made up of amino acids. That's the monomer. That's the building block. How are the amino acids linked? They're linked together by peptide bonds. And you might be thinking, oh, wait a minute. We talked about bonds earlier. There were covalent, there were ionic, and there were hydrogen. So where'd the peptide bond come from? Well, I'm sorry. You're going to find out. It seems like we're out to get you. A peptide bond is a covalent bond. But to annoy you, what we do is we give it a special name. A peptide bond is a covalent bond that connects 2 amino acids together. Why don't we just call it a covalent bond? Because that would be too easy. Just know that a peptide bond is a covalent bond between 2 amino acids. There are 20 amino acids, but there's literally thousands of proteins. So how do you get the different types of proteins? It's how you arrange these long chains of amino acids. So your amino acid structure essentially is you have, here's an amino acid and here's an amino acid, and you combine them to form the peptide bond right here. For each individual amino acid, you will always have a central carbon. On one side, you have what's known as an amino group. It has this nitrogen with the hydrogens on it. Directly opposite it will be another carbon. It's a carboxyl group with a carbon with an oxygen double bonded here and a hydroxide here. You'll always then over here have a hydrogen. And then opposite the hydrogen is what we call the R group. And that just means this is where the variable portion is. This could be as simple as a hydrogen. It could be as complex as a very detailed ring structure. So a peptide bond links those adjacent amino acids together. A polypeptide just refers to a chain of amino acids, usually at least 100 amino acids in length. And then a functioning protein is when you have thousands of amino acids linked together, and it usually does not remain as a long chain. It'll be twisted, coiled up into its unique 3D shape. When we look at the structure or shape of the protein, we divide it into 4 different categories: primary, secondary, tertiary, and quaternary. The primary structure is simply the sequence of amino acids. How does it know what order to put those amino acids in? That information is in the DNA, so it's inherited. So it's just showing here, think it's kind of like a string of beads or something. It's just what order do you put it in? That's all the primary structure is looking at. Secondary structure refers to just little small areas or regional folding of the chain. Usually what triggers that is going to be by hydrogen bonds. The 2 main types of secondary protein structures that we see are called alpha helix and pleated sheath. The alpha helix is a coiled, I often describe it as kind of looks like a slinky. So you have this twisted coil. And then the beta pleated sheet, or just pleated sheet, the beta just refers to certain structures you don't have to worry about. It's kind of the zigzag that you would see. often describe it as when you were a kid and you were hot, or even now as a big kid, that if you're hot and you have a sheet of paper and you fold it and you make a paper fan, it's that type of folding. Is it in the entire strand? No, it's just in certain areas. And you can have this multiple areas. In other words, in one long strand of a protein, that primary structure, you can have a straight stretch of the strand, and then it could go into alpha helix, and you could have a straight area. You could have another alpha helix. You could then have pleated sheets. So they're all intermixed in there. And then you end up with the tertiary structure. That's the overall 3D shape of that protein. It's either globular or fibrous. It's caused by, once again, hydrogen bonds can be involved, disulfide bridges, which is a type of covalent bond, interactions between those specific R groups. And so you have your long strand. You could have little areas, once again, where you have alpha helixes, pleated sheets, and then how the whole thing folds up. So the overall 3D shape, that is your tertiary structure. For some proteins, this is it. That's it. That's your protein. For some proteins, you can continue on and have what's known as the quaternary structure. And this is where you have subunits that must fit together to form the overall protein. So if you have multiple subunits, how they fit together now, so they're going to have their tertiary 3D shape. But then how you fit the subunits together to form the overall 3D shape, that is what the quaternary structure is going to be. So in this, there's actually, if you can, it's labeled, you can kind of see that you have 4 different subunits. And that's the quaternary structures, how they fit together. So this is just kind of a summary of your primary is the strand. Within that, where you get your individual little areas, whether alpha helix or beta pleated sheet, and then you get your tertiary structure is a 3D of that subunit, or it could be the whole protein right there. If you have subunits, how the subunits come together now forms the quaternary structure. You destroy the structure. Remember, structure determines function. So if you mess up the structure, that's going to mess up the function. How did you get the quaternary structure? Well, that was dependent upon the tertiary structure. How did you get the overall tertiary structure? Well, that was dependent on the secondary structures. How did the secondary structures form? That was dependent on the primary structure. Where did this primary structure, what determined that? That information was in the DNA. Enzymes. Enzymes are a type of protein. Enzymes are catalysts, meaning they will help speed up a chemical reaction. How do they do that? They decrease the amount of energy that you have to input initially to get the reaction started. What happens with an enzyme? Well, this darker gray is showing the enzyme. On the enzyme, you have an area known as the active site. That is where your substrate is going to come in and bind. It's very specific. If it doesn't fit, it doesn't fit. If this were a big old circle here and a square here, it's not going to fit. It just will move on. So it's very specific as to what will fit here. It's like a lock and a key. If this is your keyhole on your car, your key's the only one that should fit in there. At least that's what you're hoping. So it's very, very specific for it. Whenever it binds, one thing with chemistry, whenever you have 2 things bind together, you're usually going to see a shape change. When it unbinds, you're also going to see a shape change. So your substrate comes in and it binds. There's a shape change, and you form your products. Your products are released. When the products are released, this goes back to its original shape and can be reused again and again. Denaturation just refers to a term, the process of when A protein loses its 3D shape. If it loses its 3D shape, it's no longer functional. So it's a way of essentially destroying a protein. So you have that loss of function. How are proteins denatured? Heat is a big one. Increased heat, it will denature the proteins because it starts to unravel that 3D shape. You change the pH or you change salt concentration. Those are the three most common ways. You've all seen examples of that whenever you cook an egg. The white of egg contains protein, and when you cook it, increase that temperature, you notice it changes color, and that's because you're denaturing the proteins up. So what types of proteins do we have? We have enzymes. As I just mentioned, enzymes are a type of protein excuse me, that will be used in chemical reactions to speed them up. Some proteins act as signals for the cell. Some of them are structural components. Some are receptors that certain other things will come bind to them on the cell surface and trigger then a series of reactions. Some of them are used for transport. Some will be used for storage. Some are involved with contractile processes such as in muscle cells. And some of them, such as with your immune system, and antibodies are used as defense. So proteins are involved with just about every aspect in the cell. So we've had our carbohydrates, our lipids, and our proteins. Now we have nucleic acids. That's the fourth group of organic compounds that we're looking at. The polymers are nucleotides, buildup of nucleotides. So what is a nucleotide? As that monomer there, it is composed of three things. It will be composed of a 5-carbon sugar, either deoxyribose or ribose. It will be composed of a phosphate group, and then it will be composed of what we refer to as a nitrogenous base. This is the compound. It has carbon, but it also has nitrogen and several other elements in it. So when we look at a nucleotide, we have those three components. The way it's going to be structurally organized is you will always have your sugar in the middle, and off one end of that would be your phosphate group, and off the other end would be your nitrogenous base. Nucleic acids are designed that there is what we often refer to as the backbone of it, and the backbone is composed of repeating units alternating sugar, phosphate, sugar, phosphate, sugar, phosphate. And as I said, off the sugar will be sticking out from that backbone would be your nitrogenous base. RNA is usually single-stranded, DNA is usually double-stranded. I know some books will say always instead of usually. I put usually because as a microbiologist, I know that viruses play that whole set of different rules. There are some viruses that have double-stranded RNA. There are some viruses that have single-stranded DNA. DNA, though, usually is double-stranded, and it has the classic, what we now know as a double helix, meaning you have two strands that are going to be held together by hydrogen bonds, and they're twisted. They're wrapped around each other. And in a moment, I'll show you a structure of that. So the types of nucleic acids, you have deoxyribonucleic acid, which is DNA. It does contain the sugar deoxyribose, and its function is that it contains all of your genetic information. Everything that you inherit, everything about you is on the DNA. From your hair color to how you clap your hands to whether you can roll your tongue, everything. Ribonucleic acid is the other nucleic acid, that is RNA. It contains the sugar ribose, and this is involved in protein formation. So looking at your DNA and your RNA, if you notice here, once again, DNA is double-stranded. This blue ribbon is showing the backbone, which would be your alternating sugar phosphate components. Off of the individual sugars is where you have your nitrogenous bases, which is showing these different colored sticks, if you will, pointing off from them. The hydrogen bonds are formed from the the base on one strand between the base on the adjacent strand. And so this is going to be hydrogen bonds here holding together all of these. So if you want to visualize it almost like a ladder, where your sugar phosphate are the sides of the ladder, like what you would hang on to, and then the rungs that you would be sticking your feet on would be the bases. But then it's twisted into this double helix form. Now, RNA tends to usually be single-stranded, but same thing, sugar phosphate backbone with the nitrogenous base sticking off of it. With the bases, we'll get into this again in a moment, just know for now that A and T will always bind together. So if you have an A on one side, you're going to have a T that it hydrogen bonds to on the other strand. And then G and C, guanine and cytosine, will always bond together. Uracil is found only in RNA. So those bases, we tend to use the first letter, but just by tradition, it's always capitalized. The nitrogenous bases that you find, there are 5 of them. However, there's only 4 in DNA and only 4 in RNA. So adenine, or A, cytosine, C, guanine, G, those are found in both DNA and RNA. Thymine, or T, is found only in DNA, and uracil, or U, is found only in RNA. And you can see here the structure that all of these are ring structures. Some of them are double rings, some of them are single ring. You don't have to worry about the ring structure of the drawing it out. And then what we're going to look at, just so you know, the information, all the genetic information is found on the DNA. And then DNA will be used as a template to make RNA. And that process of making RNA by using DNA as a template is transcription. And then translation is the process where then you use that RNA as a template to make your protein. So if there's any type of mutation in the DNA, you can see where that's going to cause a change in the RNA, which is going to cause a change in the sequence of amino acid. If you change the sequence of amino acid, that's going to affect that protein structure and ultimately then possibly affect the function of the protein itself. ATP, whenever you see ATP at the cellular level, you need to think energy. And what this is you have, with ATP, you have the nitrogenous base adenine. It is connected to ribose, which is a sugar, so just like you saw with RNA. However, now over here, excuse me, what you do is there are 3 phosphate groups that are attached, and that is ATP, adenosine triphosphate. These phosphate bonds are very high energy bonds, so whenever you break them, it will release energy. So that's why I'm saying whenever you see ATP, think energy. When the cell needs energy, it will break this bond right here, removing one of the phosphate groups, leaving you with ADP, adenosine diphosphate. Now, if you have excess energy and you've got a phosphate, you can just stick this phosphate back on to store the energy. So it's your energy, once again, savings account, if you will. You need energy, break the bond. You have excess energy, put it back on. So this is constantly being broken apart and replaced.